All right, so welcome to this live class. We'll be looking at MCB 200.1, and of course, this is the introduction to general microbiology. So basically, we'll be focusing on exam likely areas that are commonly asked or commonly focused on in exams. All right. So if you can hear me, I would like you to type in the chat box below. I can hear you, sir. So we can proceed. Right, this class will last for an hour or maybe less than an hour. Okay, so I've been able to put together frequently asked questions, commonly focused areas that regards okay book vibes. Okay, good. All right, so you can hear me. That's good. So like I said, I've been able to put together areas that are most commonly focused on when it comes to general microbiology. All right, so this will be an audio class, and I just want you to pay attention. Okay, as more persons begin to join us, I would like that we have our writing materials with us, and pay very close attention, very crucial attention to everything. All right, so I'm going to be identifying some major areas that are commonly focused on when it comes to MCB exams, especially this introductory aspect, the general microbiology. All right, we have history of microbiology and the key scientists. All right, we also have microscopy and early microbial observations. We have bacterial structure, morphology, and growth. We also have bacterial physiology and transport. We have microbial ecology and enteromophiles. We have staining and laboratory techniques. We have genetics and molecular biology. We have fungi, which is also called mycology. Study of fungi is called mycology. Then we have industrial and applied microbiology. Then we have microbial diseases and pathogenicity. Then we have nutrition, enzymes, and fermentation. We've also got environmental and agricultural microbiology. At the end of this class, of course, like I said, this class is going to last till, of course, the day before your exams. And in each of the classes, we would be focusing on areas that you should pay very close attention to. Okay, so be giving you I'm going to be giving you some tips and some points to take note of. All right, when it comes to history of microbiology and key scientists. Okay, first of all, you should be able to know that the first person to observe microbiology, biological organisms, which are called microorganisms. The first person to observe microorganisms and call them by, of course, that word microorganisms was Antonio van Leeuwenhoek. Okay, Antonio van Leeuwenhoek, of course, which is of course known as the the first person to identify microorganisms. All right, so Leeuwenhoek, of course, is Leeuwenhoek. That is spelling of Leeuwenhoek. Okay, Anthony van Leeuwenhoek, he was the first person to actually observe microorganisms. Okay, was the first person, and he called them animal cools, like animal, then cules. That's what he actually called them. All right, animal cools. Then when you actually observe these microorganisms, he called them animalcules. Okay, like animal, then cules. Another thing you must take note of is the father of microbiology. Okay, the father of microbiology. Sometimes it's often credited also to Van Leeuwenhoek. All right, so it's often credited to Anthony Van Leeuwenhoek. Okay, so we also have father of immunology, also note father of immunology to be Elin Mechnikov. Okay, Elin Mechnikov. Mechnikov is spelled Metchnikoff. All right, so these things I'm telling you are the key things you need to know in the history of microbiology. They are the most frequently asked questions, okay, when it comes to the history and of course key scientists involved in the development and innovation of microbiology. All right, so like I said, you have to first of all know the the first person to observe microorganisms. I told you that is Anthony Van Leeuwenhoek, also the father of microbiology. I also say that the father of immunology is Eli. Eli is spelled Elie. Okay, Eli Mechnikov. All right, then also father of chemotherapy. Father of chemotherapy, of course, you know chemotherapy now is the study of the of cancer and tumors. All right, so or the therapy that is often used to cure cancer and all of that. All right, so the father of chemotherapy is known as Paul Ehrlich. Known as Paul Ehrlich. So Ehrlich is spelled Ehrlich. All right, so Paul Ehrlich was and has always been the father of chemotherapy. All right, so one thing that is very common, one practice that is very common in microbiology is gram staining. All right, gram staining. So the person who discovered gram staining, person who's discovered gram staining is another thing you actually have to know as well. So gram staining was discovered by Hans Christian Gram, and of course that was where the word gram came from. So the word gram came from his name. All right, I believe you can still hear me. If you're still here, if you can still hear me, say I can hear you. So proceed. So I'll be sure that my network is smooth so we can proceed. All right, if you're still here, say I can hear you. If you can hear me, say I can hear you. Okay, thank you. Ebook vibes. All right, thank you. You can hear me. All right, so now let's proceed. So like I said, you should take very important notes of these. We are looking at the key questions that are usually asked under the history of microbiology. And remember, I started by saying the first person to observe microorganisms is like a question that has commonly been asked. Okay, father of microbiology, father of immunology, father of chemotherapy. Then who invented gram staining? Who discovered gram staining? I said it's Hans Christian Gram. All right, Hans Christian Gram. So Hans is spelled Hans. All right, Hans is Hans, then Christian, then Gram. Okay, Gram is Gram. All right, Hans Christian, then Gram. Okay, so very, very important to take note of Hans Christian Gram. Then who discovered penicillin? Penicillin is another frequently asked question. Who discovered penicillin? Alexander Fleming. Okay, Alexander Fleming is actually credited to have discovered penicillin. You know, penicillin is actually a medication. So, the person who is credited to have discovered penicillin is known as Fleming. All right, Fleming, Alexander Fleming. Okay, so another one is discovery of DNA structure. Okay, discovery of DNA structure. So the person who discovered the DNA structure, there were actually two of them, all right, not just one. The persons were James Watson and Francis Crick. Okay, James Watson and Francis Crick. Crick is spelled Crick. Okay, Crick is spelled Crick. Then of course James Watson, Watson. So those scientists were actually credited to have discovered the structure of deoxyribonucleic acid, structure of DNA. Okay, so all right. So basically, who also discovered genetic transfer by transformation? Genetic transfer by transformation was discovered by Frederick Griffith. Frederick Griffith. So Griffith is spelled Griffith. Griffith. So the scientist Frederick Griffith is actually credited to have discovered gene transfer by transformation. That is how to transfer genetic information from one organism to another by also transforming this genetic information into another type or another kind of genetic information. All right, so the person who discovered that process, who postulated or brought about that process, like I said, was Frederick Griffith. All right, so one thing you need to understand now, basically in the introduction history of microbiology and key scientists, is also that microbiology evolved through discoveries. All right, through discoveries in microscopy, staining, and disease control. All right, so these three areas led to more like the bedrock of the discovery of microbiology. So different scientists came. First of all, the microscope was actually discovered because you know that microorganisms cannot be visualized using the physical eyes. All right, so the scientists first of all discovered what we actually use to view microorganisms, which is the microscope. And of course, different techniques of microscopy was actually discovered. And after that, staining. I remember I told you about gram staining. All right, how the scientist Hans Christian Gram invented the gram staining procedure. All right, and of course disease control as well, because basically most times when we make use of the microscope and all of that, will practice microbial techniques, is to ensure that microorganisms that facilitate or causes diseases are not prevalent in the environment, in order for us to actually eradicate these diseases and make the environment void of disease causative agents. All right, so some key theories were the germ theory of disease, the biogenesis, and abiogenesis, talking about how life came, how life started, the evolution of life, and the evolution of non-living matter as well. All right, so it's very important to actually understand all of this when it comes to the history of microbiology. All right, then the next area that is very very important um is microscopy. All right, microscopy. So some few core areas that has to do with early microbial observation were of course the discovery of the first compound microscope, which was discovered by Zacharias Janssen. All right, take note of all of these names. Zacharias is spelled Zacharias. So Zacharias Janssen. Janssen is spelled Jansen. All right, Zacharias Janssen. Then also what we call spontaneous generation of microorganisms. That particular theory was brought to life by a scientist called Francesco Redi. Redi is spelled Redi. Francesco Redi was the one who came up with spontaneous generation of microorganisms, more like microorganisms naturally being formed from other microorganisms. All right, microorganisms formed from other microorganisms in a generation of microorganisms. So in as much as we have generation of humans and generation of animals, we also have generation of microorganisms. So the scientist that brought about that theory of spontaneous generation of microorganisms was known as Francesco Redi. Redi is actually the spelling of ready. All right, so some areas you need to also note. Some areas you need to note are, of course, microscopy as a whole. Understand the parts of the microscope, the different parts of the microscope, from the eyepiece to the slide to the lens to different parts, okay, to the base and every other, the ocular lens and all of that. So know their locations and know their functions, understand the concept of magnification and all of that. All right. So then of course you also need to under microscopy understand the different types, okay, of microscope, understand the simple microscope, the compound microscope, and of course the difference between these microscopes and by extension the electron microscope as well. Okay, electron microscope as well. So that's it. Now then moving forward, we also have bacterial structure, morphology, and growth. All right. So um when you study the shapes of bacteria, the study of the shapes of bacteria is called morphology. All right, the study of the shapes of bacteria is called morphology. Now then we have a question that is usually commonly asked. They will say, what is the irregular grape-like clusters? Okay, grape-like clusters of microorganisms. Say irregular grape-like clusters of microorganisms is called staphylococcus. All right, so that particular phrase, irregular grape-like clusters, is very very common. If you check your past questions over the years, you see this particular term. Okay, irregular grape-like clusters. All right, staphylococcus. All right, then of course bacterial colony on solid media. Bacterial colony on solid media as well is generally known as colony. All right, generally known as colony. So then we also have what we call a bacterial wall component. That is a component of a bacterial wall, like a rigid bacterial wall component, more like it's the bacterial wall component that is fixed. Okay, it's a component, is a part of the bacterial wall that is actually fixed. When you say rigid, it doesn't move, and it is more like, will I use the word stubborn? The stubborn component of bacterial wall. So what's it called? It's called a peptidoglycan. Okay, a peptidoglycan. All right, so it's a rigid component of the bacterial wall. All right. Okay. So then the shape of bacteria, all right, is determined by the cell wall. All right, is determined by the cell wall, because basically bacterial cells are plant cells. Bacterial cells are plant cells, and they are mostly prokaryotes, okay, with a non or an undefined nucleus. So usually they tend to have the shape of the cell wall. That's how you can actually determine the shape of bacteria, by the shape of the cell wall. All right. So they are basically different things you have to also take note of when it comes to the shapes. Okay, we have the spherical shape, which is cocci, okay, or cocci, cocci. Then bacilli or bacilli is rod shape. Like I said, all microorganisms or all bacteria that you see the word cocci or cocci, like cocci, their shapes are spherical. Then bacilli or bacilli, it's like a rod-like shape. Then we also have spirilla. Spirilla is a spiral shape. All right, spirilla is a what? Spiral what shape? Okay, spirilla is a spiral shape. Then another thing, we also have what we call growth phases of bacteria. There are there are different growth phases of bacteria. The first phase is called the lag phase. Lag, lag phase. Now, this lag phase, the bacteria will start getting adapted. All right, there will be an adaptation. The bacteria will start getting adapted to its environment. Then after that, the log phase, where there will be a rapid division. The bacteria will start splitting, will start dividing rapidly. All right, then after that we'll have stationary phase, where all the nutrients will be exhausted. all the nutrients will be finished. Like in the bacteria, all the nutrients will be exhausted after, just after the rapid division. Maybe not necessarily just after, but a period of time. The period of time between the lag and the log, okay, it's usually longer than the period of time between the log phase and the stationary phase. All right. So the stationary phase, all the nutrients are exhausted. There are no formation of nutrients anymore. All right. So that's the stationary phase. And after that phase, then there is the death. The last phase is the death phase, where there is a decline of bacterial features. So the bacteria will no longer be exhibiting its features. So at that point now, there will be a decline, all right, and that's the death. So I said lag, log. Lag is adaptation, log is rapid division, stationary is of course when nutrients are exhausted, then the death phase is when there is a decline. All right, decline. All right, so then you also need to understand the bacterial physiology. All right, to understand bacterial physiology, there are also questions that usually comes under bacterial physiology. Physiology is more like functions. All right, so bacterial functions, and of course as well as the transport. Okay, so there is one thing that is called chemotaxis. Like chemotaxis is a feature of bacteria. All right, it's feature in which there is a movement towards or away from chemicals. So when there is movement towards chemicals or away from chemicals, that particular feature is called chemotaxis. All right, chemotaxis. And the transport systems that are usually exhibited by bacteria are commonly diffusion. It's either the bacteria diffuse through a surface or through a membrane, or there's an active transport. Remember what active transport is? Active transport is actually transport that requires energy. That's ATP. The passive transport does not require energy. All right, so active transport requires energy. We have primary and we have the secondary active transport. Okay, so one thing about bacteria is that for bacteria to actually move, and for a transportation of for the transportation of bacteria to actually be made prominent and active, they have to be a certain form of energy produced in the cell, in the bacterial cell. All right, so energy must be present in the form of adenosine triphosphate, which is commonly known as ATP. All right, so it's important to note that. Okay, then of course we also have the facilitated diffusion. All right, so of course in this case now we will have agents that are facilitating the diffusion process. All right, some common agents, chemicals that are used to speeding up the diffusion to ensure that these bacterial substances or bacterial agents diffuse into membranes and all of that. All right, then we also have what we call selective medium. All right, selective medium of bacteria, more like a media, okay, for culturing bacteria, is called McConkey agar. McConkey agar. McConkey is spelled MAC, then CONKEY, then agar is spelled AGAR. All right, AGAR. All right, AGAR. So McConkey agar is the selective medium, okay, for bacteria. Like I said, McConkey is spelled MAC, Mac, then Conkey, CONKEY, then agar, AGAR. So McConkey agar. Now, we have what we call motility structures. We are still talking about bacterial physiology and transport. Motility structures are more like the structures that aids bacteria to move. The structures that aids bacteria to move. All right, we have what we call the flagella or the flagella. All right, flagella. We have it moves by the aid of the flagella. All right, the flagella is more like the component part. Then we also have or more like a structure. It's a structural part that enables movement. Motility means movement. All right, then we have by gliding. It glides as well. It twitches, like TWITCHING. All right, so it actually moves by twitching as well. Okay, so that's it basically. All right, so those are motility structures, okay, for bacteria. Okay, so of course we have, like I talked about earlier, I talked about different transport types. Okay, it's I talked about active transport, and how does it move by active transport is when it depends on energy. All right, it's also moved by passive. All right, passive is the diffusion I talked about earlier. The passive transport is void of energy dependence, so it does not really depend on energy. In fact, passive does not depend on energy at all. So that's the diffusion aspect. All right, so we have, I mentioned chemotaxis. All right, so we have positive chemotaxis and we have negative chemotaxis. All right, so positive chemotaxis is the when the bacteria moves towards what is attracting it. All right, moves towards what is attracting it, moves towards the attractant. All right, then negative chemotaxis is when it moves away from the repellent or repellent. Now the repellent is what repels the bacteria, what pushes away the bacteria, so it shifts its way away from the repellent. That's a term which is called negative chemotaxis. But when it is going towards the attractant, when it's going towards the attractant, it's what we call positive chemotaxis. All right, so that's a movement strategy. That's a movement strategy for bacteria. Then, like I said, okay, we have selective medium, which is also a media type. Okay, so we also have differential, we have enriched medium. These are different media types that you need to take cognizance of. All right, so moving forward, we also have what we call microbial ecology. Microbial ecology. All right, so of course, ecology is when we are studying about living organisms and how they are getting used to their immediate environment. All right, the immediate environment is called their ecosystem. You know that. So it's actually an adaptation strategy. Remember, we talked about adaptation as the first stage. Okay, as the first stage, when we are talking about growth phases, I said adaptation is under the lag phase. Log is rapid division. Don't forget that. I also mentioned stationary phase, which is when nutrients have been exhausted, and of course death when there is a decline. All right, so don't forget that. Okay, so then I also mentioned that okay, moving forward, I just talked about microbial ecology, right? So it's more like microorganisms, when microorganisms are beginning to get used to their immediate environment, and of course trying to survive under extreme conditions. All right, so when microbes are actually surviving in extreme conditions, they are called extremophiles. Extremophiles, from the word phils is spelled Philes. Philes. Philes, feels comes from the word philia. Philia has to do with love, philia has to do with attraction, philia has to do with affinity. All right, so when microbes are actually surviving in extreme conditions, they are called extremophiles. All right, because they actually love conditions that are extreme. All right, there are some bacteria that will not survive in extreme conditions. All right, there are some bacteria that will not survive in extreme conditions. So it depends on the kind of extreme conditions. There are different types of extreme conditions. We have the ones that survive in cold environment or cold conditions. Okay, they are called cold loving bacteria. So they are called psychrophiles. Psychrophiles is spelled Psychrophiles. Psychrophiles. All right, so when bacteria is actually in a cold environment and it is surviving in such kind of condition without even shrinking or without even undergoing any form of stationary or decline state, then of course it's called a psychrophile. We also have the ones that actually survive in high pH environment. pH that is of course in alkaline form. All right, in between 8.5 and 11.5. All right, they are called alkalophiles. Alkalophiles, from the word alkaline. Alkalophiles, which means that they love alkaline environments. All right, so That's another form of extremophils. Okay, then we also have what we call the halophils. Halophils is spelled Halophils. Okay, halophils are, of course, those bacteria, those microorganisms that actually survive in high salt environment. So they are called halophils. All right, they're called halophils. So when we talk about all of these, we are focusing on adaptations. All right, so getting used to this, their environments, adaptations, and of course adaptations includes, of course, first of all, enzyme stability, because for a particular microorganism to actually get used to a particular environment, the enzymes that are facilitating the reaction there has to be stable. Okay, it's not going to be an inconsistency in enzyme action. Okay, it has to do with a stability in enzyme action. And of course, the membranes have to be modified as well. Membrane modifications is another key aspect of adaptations. Like when I talk about membrane, I'm talking about the cell membranes. All right, cell membranes, and of course not necessarily cell membranes of the bacteria themselves. Okay, cell membranes of any organism that they are actually diffusing into. Okay, those cell membranes of those organisms have to go through modification and processes every moment. All right, then of course the environments are commonly hot springs environment. We also have deep sea environments where you commonly find these bacteria. All right, where you commonly find them. Then we also have acidic and alkaline conditions. All right, acidic and alkaline conditions, those are the kind of environments where you would actually come across these microorganisms and all of that. All right, so okay, so basically that is it. Okay, so we also have the staining and all of that. The staining is one other thing you need to actually take note of when it comes to gram stain. Gram stain is known as the most important differential stain. Very, very important. Don't forget that it was discovered by a scientist that we talked about initially, and that scientist was Hans Christian Gram. Don't forget that name. All right, so it's the Gram stain is the most important differential stain, because it can be able to cut across all kinds of microorganisms when you want to stain them in a differential media. All right, so another question that is commonly asked is questions concerning antiseptics. All right, they will ask you, purpose of antiseptics is what? Is to prevent contamination. All right, so it's to prevent contamination. That's the main purpose. All right, of that's the main purpose of antiseptics. All right, is to prevent contamination. All right, so that's it basically. All right, then we have what we call hemolysis as well. So there are different forms of hemolysis when it comes to microorganisms. All right, we have what we call partial hemolysis. All right, partial hemolysis is the alpha hemolysis. Okay, we have beta hemolysis. Beta hemolysis is complete hemolysis. Then gamma hemolysis is no hemolysis at all. All right, so when there is no hemolysis has to do with a breakdown. Whenever we do breakdown of more like the blood components in microorganisms, and when you hear the word hem has to do with blood. All right, then lysis has to do with breakdown. All right, lysis has to do with breakdown. Okay, so it's very important. So in that gram staining I was talking about, when you stain, okay, gram positive is usually purple. Okay, then when you stain, gram negative is usually pink. Gram positive is purple. Remember, we have gram positive and gram negative. So the gram positive is usually purple in color, while gram negative is usually pink in color. All right, so it's very, very important when it comes to classification and diagnosis of diseases. All right, very important for classifying diseases and actually diagnosing them. All right, actually diagnosing them. So it's very important to note all of that. All right. So one thing you should actually bear in mind is that if you've been able to look at questions that are commonly asked, you realize that one thing that is common about these questions is that they tend to be related to specific topics. All right, and these are the topics that we've been able to touch so far. So like I said, okay, we'll also proceed, we'll also continue with some other topics, but don't forget that you have to study about the staining and laboratory techniques. All right, any question you see on it, answer. I've already given you giving you a heads up. Okay, microbial ecology as well. I told you about microbial ecology and of course extremophils, the different types. I mentioned everything about bacterial physiology and transport. I've also talked about bacterial structure, morphology, and growth. I've talked about the growth phases. I've talked about microscopy and early microbial observations. Okay, I've also talked about the history of microbiology and of course key scientists that played various roles in the development of microbiology. Now, there's one other aspect that you must not avoid, and that is the study of fungi. And fungi is called mycology. So the study of fungi is called mycology. All right. So then fungal infections. Okay, fungal infections are generally known as mycosis. Mycosis is spelled Mycoses. All right, Mycoses. All right, it's that's what we call mycosis. All right, I don't know, can you still hear me? If you can hear me, say I can hear you, sir. All right, let me be sure that the network is smooth. If you can hear me, say I can hear you, sir. So we proceed. If you're still here and you can hear me, say I can hear you, sir. All right, good. Okay, nature. Okay, your girl nurture. All right, good. Who else can hear me? Who else can hear me? Chukuda Goodness, are you there? Good news, rather. Are you there? Okay. Good news, if you're there, if you can hear me, say I can hear you. Bright Faith, ebook vibes, are you still there? All right. Good. All right. So impressive. Now let's proceed. I was talking about fungi. Okay. I say that the study of fungi is called mycology. Okay. Study of fungi is called mycology. All right. So and fungal infections are generally known as mycosis. Mycoses. Mycosis. All right. Then we have what we call yeast. Yeast is, in fact, fungus. In the kingdom fungi, the organisms that tends to be more commonly studied are yeast, mushroom, and bread mold. Mould. Okay. So basically they are all unicellular. Most of them are unicellular. Let me just say most of them are unicellular. Just a few are multicellular. So it means that for instance now yeast is actually a unicellular fungi. All right, yeast is actually a unicellular fungi. It grows by budding. Budding. It's formed by budding. All right. So when fungi has this ability to switch forms. It can switch its forms from one form to another. Okay, so that process is called dimorphism. Dimorphism, dimorphism. So dimorphism is actually the ability for fungi to switch forms. Like it can be like this one time, the next moment it's another way. All right, so the ability for it to switch forms is known as dimorphism. All right, so we also have fungi too, having affinity for the roots of plants. Sometimes you see fungus at the bottom, at the base of the roots of plant. You see mushroom growing at the base of a tree.
or at the base of one plant like that, you see mushroom there, you see yeast there, or maybe you see mold there. Okay, so that is usually called, when fungus and plant roots combine, it's usually called mycorrhiza. Mycorrhiza is spelled Mycorrhiza. Mycorrhiza. Okay, Mycorrhiza. Mycorrhiza. All right, so it's when fungus um combines with plant roots. So it's more like a kind of relationship between fungus and plant, the roots of plants. All right, so when fungi or fungus also have a form of relationship with algae, okay, they are called lichens. They are called lichens. Lichens. Okay, they are called lichens. Some people pronounce it as lichens. Okay, but it's usually commonly pronounced as lichens. Lichens. That is fungus plus algae. It's called lichens. All right. So we also have on the cell wall of fungi, we have what protects the cell wall. Okay, the cell wall of fungi is usually protected by a polysaccharide called chitin. Okay, you know polysaccharides now. Polysaccharides are, of course, carbohydrates that have multiple sugar molecules. That is poly sugar molecules. That is numerous sugar molecules. All right. So when we have the cell wall of fungi is usually protected by chitin. 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 So it's a polysaccharide that protects the cell wall of fungi. All right. So that's something that's very, very important for you to take note of. All right. So you need to know that the fungi are commonly known as decomposers. All right, that's the kind of nutrition, that's the kind of nutritional feature that they have. Generally they are saprophytes. All right, which makes them decomposers. You know, under nutrition we have the autotrophic and the heterotrophic, right? Under autotrophic we have the chemosynthesis and photosynthesis. Then now coming down to heterotrophic, that's where we have holozoic, symbiotic, saprophytic, parasitic. So you know holozoic is the one that we actually exhibit. We have scavengers, we have predators and all of that, blah blah blah. Symbiotic is that mutual, called, you also called mutualism, mutual relationship. Okay, mutual relationship. So that symbiotic relationship where both organisms are actually benefiting from themselves is only common only when there is a feeding relationship between fungi and algae that I called leaching. But generally, yeah, fungi actually exhibits saprophytic, okay, which makes them generally decomposers. Which means that they usually have the ability to decompose substances. That's why you see something like mold, bread mold on breads that have stayed for long. Bread will don't spoil. You go come they see white things on top. All right, that's bread mold. You see mushroom in areas that maybe water has actually entered a particular wood and all of that. You see mushroom growing from there. You see mushroom also in waterlogged areas, just close to like the roots of trees that I actually call the mycorrhiza. Okay, where you find fungus and plant roots. All right, so they are generally decomposers. They are generally decomposers, which of course made me to actually say that they are saprophytes. All right, so they exhibit saprophytic mode of nutrition. All right, so but basically they have importance. So in case you see a question, what is the importance of fungi? They are important in fermentation. Okay, when it comes to fermenting, all right, they are very important in fermentation, very, very important in fermentation. Okay, you can use something like yeast to ferment. Okay, You can use something like yeast to ferment. Okay, they are also used to produce antibiotics. Yes, they are used to produce antibiotics. Okay, they are also used for food production. They used for food production. Okay, there are some food substances, there are some food products that are actually produced by using yeast. There are some that you see some fragments. They will say they use some fragments of a certain proportion of mushroom and all of that. These are just the ones that I mentioned because they are the ones that are commonly known. Okay, but there are different varieties of fungi. All right, there are different varieties of fungi. Okay, so basically that's it. Now then we also have what we call applied microbiology. Industrial and applied microbiology is another area of microbiology that um it's very important to note, to be familiarized with when it comes to this general microbiology, which is of course MCB 200.1. Now um okay, so basically, what is industrial microbiology? Industrial microbiology is the process of using microbes in the industry. When you're using microbes, when you're using microorganisms, when you're using microbial agents in the industry, that is industrial microbiology. All right. Then in when we come to when we talk about applied microbiology, is the more like the application of microbiology, the techniques that facilitate the application of microbiology. We have bioremediation. I believe you must have heard of bioremediation. Okay, so bioremediation is actually the process of removal of pollutants. So when pollutants are actually removed from a particular environment, okay, that's what we call bioremediation. Okay, removal of pollutants is called bioremediation. All right. Then we also have biofuels. Okay, also have biofuels. So biofuels are actually gotten from organic matter, and these organic matter are also components of microbes. Yes, microbes are in organic matter as well. Okay, so we have biofuels. We also have what we call biomethane. Okay, you can be asked what is biomethane and all of that. Biomethane is actually a substance that is used for anaerobic digestion. All right, anaerobic is, of course, does not require the use of air, right? We have aerobic and we have anaerobic. Okay, so biomethane is actually a form of applied microbiology that enables the use of substances like this biomethane to perform anaerobic digestion. That is, of course, in the absence of air and other gaseous substances. All right. So in applied microbiology, we also have biopesticides. All right. Biopesticides are actually agents that are used to control pests. All right, to control pests so that's biopesticides. All right, so generally we can say that microbiology is applied in food industry, is applied in in energy, that is biofuels I just mentioned. It's also applied in the environment for waste treatment. Okay, which I talked about which I talked about removal of pollutants, which is bioremediation, waste treatments. So microbes are used for fermentation. I mentioned that, and also there's another one that you need to understand that is very important to note. Microbes are also used for enzyme production. Yes, enzymes can actually be produced from microbes. Okay, now enzymes are general are proteins by nature. They are naturally proteins, right? Good. And proteins are usually formed from the building blocks of amino acids, right? So these amino acids most times are commonly gotten from certain microorganisms. Yes, because amino acids are like um the aside the fact that they are the building blocks, they are also the byproducts, right? They're also the byproducts these these proteins that we talked about. So it's very important to actually note that there are certain enzymes that can actually be produced, okay, from microbes, all right, through the different culturing techniques. Enzymes can actually be produced from microbes, all right. So we also have what we also need to focus on microbial diseases. Very crucial to focus on microbial diseases. So microbial diseases, of course, are diseases are that are actually spread through microbes, right? Through microorganisms and all of that. So one microbial, it's one group of microbial disease that is usually very prevalent is the ones that are caused by pathogenic bacteria. Okay, you see every time people are taking antibiotics and all of that. Most times antibiotics are antibacterial, all right. So but they are caused by pathogenic bacteria. Most microbial diseases, don't forget that, are caused by pathogenic bacteria. So one thing that is common about this pathogenic bacteria is that once they invade, they first of all invade. There are three steps. There are three steps in these diseases being prevalent. They invade first. The pathogenic bacteria invade. After invading, they survive. Then they spread. They invade, then they survive, then they spread. They cannot spread if they did not survive. So sometimes when if the body system is being invaded by bacteria, okay, there are some chemicals that tends to repel. Remember what I talked about chemotaxis, right? So there are some chemicals that tends to repel these microorganisms. All right. So what happens is that they move away. That is negative chemotaxis. They move away. All right. But if the chemicals cannot have the ability to repel these bacteria, what happens is that they now move towards it, and it will now be a form of attraction. And what will happen is that instead of declining, instead of dying, what will happen? They would survive, and after surviving, they will spread. That's why you hear cases that bacteria have actually spread in a particular region of the body and all of that. Okay, so I believe you understand. So basically, when they invade, they have to invade first, and for them to spread, they must survive. So it's three steps: invade, survive, and spread. Those are the three steps that pathogenic bacteria usually employ. Okay, when it comes to generating microbial diseases. All right. So in order for you to actually prevent these pathogenic bacteria from spreading, if they eventually succeed in invading, they must not survive. They must not survive, because most times once they survive and they spread, it's difficult for them to actually decline. I know what I mean by decline. Decline means death, right? So the best stage to actually prevent them is the environment, ensuring that the environment is void of this bacteria. And even if the environment is not void of this bacteria and they invade, they should not survive. All right, they should not survive. They should be able to they should be eradicating techniques or eradicating patterns to ensure that they don't spread, all right, after surviving. So we have common diseases, common microbial disease. We have the Bordetella pertussis. Okay, Bordetella is spelled Bordetella. Pertussis is Pertussis. Okay, Bordetella pertussis is what we know as whooping cough. All right. Then we have the Candida albicans. All right. Candida albicans is called, of course, fungal infection. When someone has a fungal infection, okay, it's called Candida albicans. All right. So we also have Aspergillus. All right, Aspergillus, or as some people pronounce it as Aspergillus, but it's usually Aspergillus. So this one is fungal disease. The difference between an infection and a disease. All right. So more like an infection tends to affect a particular site. All right. An infection targets a particular site. but a disease tends to spread more than an infection. Okay, a disease tends to, and most times diseases tends to be more communicable than even infections. All right, so we are that's the point. All right, then we have also the dermatophytes. All right, dermatophytes are skin infections. All right, so these are common microbial diseases. There are many more. Okay, and many more. So you have to understand that all of this spread is under the whole idea of pathogenicity. Pathogen is of course disease, right? So genicity is how these diseases starts and how they spread. How they start and how they spread, and the starting is not just by starting, but they have to grow, they have to invade, they have to survive, they have to now spread. Okay, they have to now spread. So basically, the this old spreading can be invasive. Okay, can be invasive. They will invade. It's called invasion. They will invade. Okay, they will invade the particular site and infect that site during their spreading process. Okay, so we have what we call types of infections. All right, so types of infections, we have localized infection and systemic infection. The localized infection is the one that focuses on only one area. It can focus on a cell, a tissue, or an organ. A cell, a tissue, or an organ. But the systemic infection affects the entire system. For instance, if there is an infection in the gastrointestinal tract, it will start from the mouth. It will infect the pharynx. It will infect the esophagus. It will go down to the diaphragm. It will enter the stomach. It will enter the pancreas. It will enter the spleen. It will enter the gallbladder. It will enter the liver. It will enter everywhere. Small intestine, large intestine, and the rectum, the anus self. So it will infect the whole, and most times systemic, you see systemic infection, systemic infection is fatal. Okay, it's fatal. It's the one that sometimes the whole process of healing and all of that is not usually easy. All right, so just if you can hear me, please indicate that I can hear you, sir. All right. So let's proceed. Okay, you say you can hear me. Okay, so you can hear me. All right. So let's proceed. All right. So moving forward, I was talking about this pathogenicity. All right. So
like I said, there are of course types of infections. I say some can be localized, some can be systemic, and the ones that are systemic can actually be very very difficult to go through curing and all of that, because before you would do um, the whole curing of the entire organs, okay, the whole curing of the entire organs, at that point in time you you would already experience, in fact you already see that that particular individual has gone through a lot of gone through a lot of damage already in that particular system. And most times, like I said, it's fatal, so it usually leads to death and all of that. All right, so it's very important to note that. All right, so when we talk about microbiology, is also very important in nutrition as well. Like I told you, fermentation is one thing that your microbes can actually be used for. One thing microbes you are used for, when I mention microorganisms, please don't bear in mind that microorganisms must be harmful. Microorganisms must not be harmful. All right, they are dangerous ones, they are the ones that are also safe. All right. uh so fermentation improves taste, it improves aroma, it improves flavor. All right, most of the time we use microbes to actually improve the flavor. We use microbes to also preserve our food. All right, we use microbes as preservatives. All right, so most of our preservatives are produced using microbes. So it's actually very important. Then we also have the, remember I told you about enzymes that these guys, which are the microbes can actually be used to produce enzymes like amylase, protease, and pectinase. These are common enzymes that we know, especially these are commonly digestive enzymes. All right, these digestive enzymes are usually used to facilitate the digestion of certain nutrients. All right, so they are commonly produced by these microbes. All right, they are commonly produced by these microbes. All right, so it's very important to actually note that. All right, you see your yogurt, your wine and all of that. Bread self is, they use yeast is being used to produce bread. All right, and yeast is a fungi, and fungi is a microbe. All right, wine production as well. Okay, yogurts as well, like I said. All right, so these are the areas where they actually show a lot of nutritional benefits. Okay, so it's very 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 important to understand that. Now, so, for some tips, there's some tips that I must give you, that you must take note of, that are very very important. You see definitions, very common definitions, all right, of microbial terms. All right, please don't play with them. See those definitions, you see scientists and discoveries, you definitely see them in exam. Okay, scientists and discoveries, don't play with them. See some processes like I mentioned, gram staining and fermentation and growth curve, where you have the log, the lag, the stationary, the decline, which is the death. That growth curve, also understand how the different processes undergo, how it moves from one process to another and all of that. All right, then of course applications too, I've talked about. I mean, both on industrial and medical, try and ensure you revise the applications of microbes. Okay, so that's it basically. All right, so test yourself, ask yourself questions, study past questions, answer past questions. All right, check check your retention ability. Okay, to that is your ability to retain information. Ensure you upgrade that as well, and all of that. So we'll still be having this class tomorrow. Okay, we'll still be having this class tomorrow. It may be at the same by the same time. Okay, most likely at the same time that we had today, within 8 9. All right, so just feel free to tune in. Tomorrow we'll be having our physical class, of course, on chemistry as well tomorrow. But this MCB 200 will be online. Okay, I will be dropping the YouTube link as well, like I did for today. All right. So once you get the YouTube link, just tune in and have a good time. Okay, have a good time. If you have any questions as well, feel free to always ask. You can also text me via WhatsApp as well. Okay, if you have any questions whatsoever on whatever I have taught. All right. So that is all for tonight. This is where we'll wrap the whole session and we'll draw the curtain for the night. So thank you for tuning in, and I would send you the YouTube link for tomorrow's class. Don't stop reading, don't stop studying, don't stop trying to gain knowledge. All right. So yes, of course, we will do what different from what we did today. Of course, tomorrow we'll do a different thing altogether. It can't be repetitions. All right, it can't be repetition, but ensure you revise what you've learned today. All right. So tomorrow we'll still meet again. All right. So um, see you tomorrow. Bye. All right. Someone is asking if this class can be sent as a PDF. What PDF now? We're having an online class. This is an online class now. What PDF? Watch the video. I don't usually send PDF so much because sometimes you send PDF and students don't go through it. So this particular session you can actually go back to the beginning and listen to it again and take down your notes again. All right. So there is no need for any PDF. I'm not using any PDF in teaching you. All right. So I have all of these things already. I have all of these things. I've noted all of these things. I have them upstairs already. So I'm just giving them to you. So that's how we also do tomorrow. Okay. I'm not dropping a PDF. I will not develop any PDF on this. Attend the online class and pay attention. Most times I drop PDF, students don't go through it, or students tend to go through it the night before exams, and that doesn't help a student. The student who must achieve academic success must ensure that he or she studies early enough when it comes to any course whatsoever. All right, not when it's the 11th hour. So like I said, we'll still have this class tomorrow. Take care and good night, everyone.